Good evening, all the centers. Shillong, good evening. Everybody. Yes, children, how are you? I'm having a bad throat today. Hope so. My voice is clear to one and all. Is it? Okay, how are you, children? Nice? Tired? Back from school? Okay, I understand. Okay, fine. So, we completed a chapter in the last class. We did excretory waste and its removal, isn't it? So, do you have any doubts from that chapter? Everybody? Do we have any doubts? Let me know, please, if you have any doubts from that chapter. Yes, children? Do we? Yes? No? Nothing? No doubts? Seriously? Okay, fine. So, I've started sending you assignments, isn't it? You must have received. Or if you are back from school, you will go and access your mobile at home now. You will be seeing some of the assignments I have sent in the morning, first half. So, you are supposedly going to do those assignments. Okay, so class also I will be giving you an assignment that will be class wise, and we will try to have some questions according to your board formats. So obviously this year is not your board, but yes, according to board format means the type of questions, the number wise, like what kind of questions can come for 1 marks, for 2 marks, for 3 or 5. Those type of questions we will start doing from today itself, from now itself, so that we can have a good grip on everything. Obviously for a school exam, as well as we will know to write how much is sufficient for our boards. Like if you are writing a long answer type question, I would be expecting you to write 120 words minimum. That is for 5 marks. So if you are writing for 1 marks, no need to write a big paragraph. You just need to write a few points, a few words, or what you can do is that you can just write it down in a single or 2 lines. That's maximum we actually suggest everybody to write for 1 marks. For 2 marks, it should be for 40 words or 40 to 50 words approximately. And for 3 marks, it would be 60 to 75 will do the job. So you have to support the answers wherever possible. Obviously, if the question demands for a diagram, you have to draw the diagram well labeled. And if possible, for 3 marks and 5 marks, you can additionally give examples, as well as, if possible, if the diagram is not too lengthy, you can try to. If the diagram is not too lengthy, that is a given point, you can try. If it's small, if it's just you can explain with the help of diagram, you can try. Okay. So, in that ways, we will be working out. Yes, hello. In that ways, we will be working out and we will be doing, try to do, obviously, you and me both, will try to do something great. So, I guess we were supposedly going to start biomolecules in today's class. Is it? Isn't it? You will told me to start biomolecules. So, we have completed somewhat, 3 chapters, that is digestion, breathing, 4. Digestion, breathing, body fluids, and excretion. We have done 4 chapters in physiology. Now, I will be moving back to the first phase, that would be the biomolecule section. Since biomolecule is a part which will be common to both botany and zoology. So, we will be having an access to those kind of things that is also found in the plants as well as in the animals. So, biomolecule will be a common chapter, just like cell and cell division. Obviously, cell and cell division will be taken up by Vineet sir. So, I will be teaching you from that section the topic called as biomolecules. Let us see what biomolecules are. Okay? So we are going to start up with biomolecules. So first of all, bio means what? Something which is present in living. And molecules, obviously, the type of substances found in living organisms. So if we try to classify the molecules of the biological world, of our world, we'll be able to classify the things into two categories. So either it would be the biomolecules, either it would be organic, or it would be inorganic. So we classify the biomolecules in two sections. What are they? Either that has to be organic or that has to be inorganic. So now what is organic? Obviously, they are the compounds having C, H and O. Carbon, hydrogen and oxygen, all the three should be present in the same molecule. We call them to be biomolecules. Whereas inorganic, they contain metals and non-metals found in living world. So whatever living world has in the form of metals and non-metals, we will call them what? We will call them to be inorganic. So compounds having carbon, hydrogen and oxygen, obviously they are said to be organic compounds. So there was this differentiation created. People tried to make differences. They started to do creation of differences. They started to show you differences in this way. First thing. Then there was a need. The need arose to differentiate on the basis of how we can separate them. Okay. So if I am giving you a whole, suppose, liver of an animal. Just take my example. Liver of an animal, and I am telling you that just let me know the organic and inorganic components among them. So what are you going to do for that? So let's see. To separate out what procedure was initiated, how it was done. So first and foremost thing, separation process for organic was very much separate from the inorganic. So first organic, what we did was, we will take the tissue, we'll add trichloroacetic acid. We'll call it TCA. Trichloroacetic acid or TCA we are going to take, and we are going to crush this in mortar and pestle. You must have seen mortar pestle in your chemistry labs. It's a kind of instrument, you can say, which is used to crush things. There will be mortar and there will be a pestle. We use this thing to crush them. So we are going to crush it in mortar and pestle, and this is going to form what? Forms a thick slurry. Slurry means a paste-like composition. So this slurry, you need to filter it. Slurry is filtered. By using a muslin cloth. Okay, you're going to just filter this using a muslin cloth. Did you copy, children, up to this? Can I move ahead? Everybody. So what we did, we took the tissue. I'm having a really bad throat today. Just excuse me for that. It can be a liver or it can be a plant part. It can be anything. So we are going to take it and we are going to just make a paste-like composition by mixing it or just crushing it in trichloroacetic acid, TCA. Yes, Shillong, am I clear up to this? Can I filter it now? Can I filter? Yes, how many of you are present? Just give, send your attendance, please. Are we clear? Just give me a thumbs up. Will not make the procedure so long. Are we clear up to this? Okay, fine. So thick paste is, okay, very nice. So this thick paste is what? This thick paste is filtered. Now, let us set up an apparatus for filtration. Sorry. What is this? Yes, what is this? What is this? Very good. And what is this? So we are placing a funnel over the filter and trying to filter the things. So obviously we are going to get what? We are going to get two phases. Simplest example, when you filter tea, you get the tea leaves over here. Now this, in chemistry, you will learn to tell residue. But over here, I will be calling it retinate for a particular reason. And this section is said to be filtrate. So what we did, again, same procedure, tissue crushed in trichloroacetic acid. Then you are going to filter it using a muslin cloth. After filtration process, you are going to get two phases. The one which will remain in the particular muslin cloth called as retinate, similar to a tea leaves, what we get after filtration of tea. And the second is filtrate, the second section. It will be tea that we use to consume. So you are going to get two sections, the retinate section and the filtrate section after the whole procedure. Just have a pause on this particular part. This is a separation procedure. We'll just pause over here. Okay, I'm going to explore this thing next to it, but just pause for some time and we'll see the separation process for inorganic. So inorganic separation procedure is a bit different. What you are going to do over here, you are going to dehydrate the tissue first. Dehydrate means you are going to remove all the water present inside the tissue. You are going to dehydrate it. After dehydration of the tissue, what we are supposedly doing, we are going to burn this. So the tissue is burnt after dehydrating, and after burning, what we are going to get is ashes. Now these ashes will be subsequently used for the salt analysis. For analyzing different types of salt like sodium, potassium, phosphate, sulfate, magnesium, manganese, and etc. So this is a particular procedure where we do for separation out of organic and inorganic phases. So if you are getting an organic thing, you are not going to dehydrate it, you are just going to mix in trichloroacetic acid and crush it. If you are going to get the inorganic phase, you are going to just dehydrate it and burn it. Am I clear? Yes, everybody. Excuse me. Yes, children. Clear, sorted? Yes. Can I see thumbs up? Okay, very good. Yes, Shillong. Tell me, children, are you clear with this? Can I move ahead? Okay, fine. Just let me know in case of any confusion. So now you're going to get what? Sorry. Okay, I will just take a liver or a stem or a leaf. Okay, now I have given you a liver or a particular plant part just like leaf, and I am asking you to tell us the organic composition of this particular organ. Now what you will be doing is that you will be taking the tissue, mix it in trichloroacetic acid and start crushing it. Crushing means just trying to make a thick paste-like thing. Then this thick paste will be called as slurry. The slurry will be filtered by somewhat this kind of setup. Now, something which you get on the upper part, which is not coming down to the filter, will be said to be retinate fraction. And the particular composition coming downwards, filtered, is said to be the filtrate section. Clear? Yes, Shillong? Clear, children? Everybody? Now, I am moving. So what we started to do, we did somewhat salt analysis over here, isn't it? We did salt analysis over here, see? We did salt analysis over here. This thing. So you have to analyze the retinate and filtrate also. So similarly, what scientists at that time did, they started to analyze the components of the filtrate and retinate. So I'm just making two sections, filtrate and retinate, and let's see what were the components present over there. So when analysis of filtrate component was done, it was found that there were substances which were So there were substances which were soluble in the trichloroacetic acid. So we call them to be, the filtrate section to be acid soluble pool. Now similarly, This was having the components insoluble in TCA, so called as, what it would be? Can I call it acid insoluble pool? Pool means fraction. Obviously, this would be called as what? This would be called as acid insoluble fraction or acid insoluble pool. So what we are having? We are having two pools, two sections. One is acid soluble and other is acid insoluble. Now we did further analysis and what we found was this contains molecules with a molecular weight of less than 10,000 Dalton. So whatever is less than 10,000 Dalton of molecular weight will appear in the filtrate section. And what does this contains? Obviously, all those substances which would be having a molecular weight of more than 10,000 Dalton. So if it is more than 10,000 Dalton, what it would be? It would be in the retinate fraction, and if it is less than 10,000 Dalton, it would be in the filtrate fraction. Okay? Clear, children? So on this basis, can I say this, that the filtrate part has bio micromolecules? Micro means small. Why? Because it has a molecular weight of less than 10,000 Dalton. And this has bio macromolecules. Why? Because it has molecular weight of more than 10,000 Dalton. Clear? Tell me something. Yes, no? Thumbs up. Still copying? Okay. Yes, children, tell me. Am I clear? Okay, very good. Now, further analysis was done, like just in case of inorganic section, what we did, we saw Na plus, K plus, the peculiarities. We saw the typically the particular sections name-wise. Similarly, we want to know about filtrate and retinate fractions also, that what are the components present, isn't it? So what we did was, we started to analyze it. So what analysis gave us, this has amino acids, this has monosaccharides or simplest carbohydrates. Okay? This amino acids and monosaccharides, this has nucleotides. And what they have? The retinate fraction, the acid insoluble part, was found to have proteins. They're found to have complex carbohydrates. They are found to have nucleic acid, that is DNA or RNA, and lipids. Now, lipid was something which was very, very special in this fraction. The reason being that the lipid was having only a molecular weight of 18 to 800 Dalton. The molecular weight of lipid is very less. How much? It is only 18 to 800 Dalton. But the molecular weight of these three substances, if you take into account, of these three, is more than 10,000 Dalton. So what could be the reason that lipid, in fact lipid should have been come in the micromolecule part, because what we did now, just now, we did this thing, isn't it? That the molecular weight of micromolecules is what? It is less than 10,000 Dalton. Isn't it? See this thing? And the macromolecule section was what? It was found to be more than 10,000 Dalton. So according to the molecular weight phenomenon, lipid must have appeared in the filtrate part, in the acid soluble fraction. But instead of appearing here, it is appearing where? It is appearing in the macromolecule section. Why? Question is clear to one and all? Question is clear? Yes. Any idea? No idea. Yes, Shillong. Tell me, children. Tell me something. Are we connected? The question is clear to everybody. Yes. The question is clear? Yes, Shillong. Nothing. First tell me, are you clear up to this? Are we clear up to this? Just tell me. Ah, good. Nice. Almost. Yes, very good. Okay, it's insoluble in water. Very good. They are fatty acids. They are components of fatty acid, in fact. Okay, fine. Very good. Nice one. So, my question is that lipids have a molecular weight of only 18 to 800 Dalton. But still they appear in the retinate or acid insoluble pool. They should have appeared in the filtrate section. Because everything in filtrate is what? Everything in filtrate is less than 10,000 Dalton. See over here. Everything in filtrate is what? It is less than 10,000 Dalton. So it should have appeared in the filtrate section. But still it is appearing in the retinate section. So if a question like this comes to you, you have to answer. Very good. Good attempt from your side. So we'll just modify a little bit. That very good. Lipids are insoluble in trichloroacetic acid. The first point. That is, they are insoluble in trichloroacetic acid. Next point. They form. What is sir teaching you now? In the botany, what is sir teaching you? Is he teaching you cell? What is sir teaching you, children, in botany? Yes. One of you please get up and type, what is sir teaching you in botany? I wish to know, because if it is he is teaching cell, that would be an easy thing. Yes. They form. Okay, Protista is teaching. So, sir must have told you that all the organisms, they are having a membrane, cell membrane, isn't it? So, what is lipid forming? They form cell membranes. In the cell, and also the coverings of cell organelles. So, when you crushed them, remember you crushed them in TCA. The cell membranes got ruptured. The cell membrane was ruptured. And lipids started to get disorganized. So the organized structure of the lipid, they started to break down. They started to rupture. Started to get disorganized. Now this disorganized structures of the lipid, all these disorganized structures of the lipids, they actually tried to form a bond in between. So lipid particles or the molecules started to form a hydrophobic bond among themselves. Now what is a hydrophobic bond? Over here you can see what kind of bond they are forming. They are forming a hydrophobic bond. So a hydrophobic bond is a bond that keeps it away from water. Phobia in science means fear. So this is a particular bond which is water fearing. And this helps the lipid molecule to keep away from the water. So they formed a bond in between which actually made them keep a distance from the water. So this bond formation was in such a way that if this is a single particle of lipid, suppose these all are single particles of lipid weighing 18 to 800 Dalton each particle, they started to get arranged in this format. Okay, and this red structure in between is the bond formation of these structures. So this particular structure is now called as what? Now called as missile. So all these lipid particles, these are what? These are lipid particles. They started to form a hydrophobic bond, and after the formation of hydrophobic bond, the structure was somewhat round and called as missile. Now just imagine a situation, you have a door in your classroom. Can you see? All of us have a door in our classroom. If you try to go out of that door alone, you will be able to do so. But if you just make a circle, holding hands together, all of you, all the children who are present in the class, all of you start to form a circle, holding your hands together, would you be able to pass through a door outside? Would you be? Just try to hold hands together, everybody form a big circle and try to go out from the door as such. Will you be able to? Yes, tell me, no, yes. Would you be able to do? Would you be able to go out of the door when you form a big circle, holding hands together? Yes. Sorry, I cannot see you people clearly. No, obviously, yes. You won't be able to do so. Similar is the case with the lipid also. Lipid was 18 to 800 molecular weight alone. But when it held hands with other molecules of its own nature and formed a missile, that is a particular round structure, and tried to go outside the pore of the muslin cloth, it failed to do so. And this was the reason why it remained in the retinate section or in the acid insoluble part instead of passing down in the filtrate section or in the acid soluble pool. Let's do everything once again. So first of all, you need to take care of this thing that lipids are insoluble in trichloroacetic acid, first and foremost thing. And where is lipid found? In the cell membranes and in the cell organelle coverings. You have to now crush the lipid, the cell in TCA. When the cell was crushed, when the organ was crushed, the cells, they also distorted, the cell membranes ruptured, and the lipid started to get disorganized. Then, what lipid saw near to it? It saw TCA. It was very, very scared to see TCA. What it did was, it started to form a hydrophobic bond among themselves, and the particular structure called after the formation of hydrophobic bond was called as missile. Now this missile, let's see, was unable to pass So it was found to remain in the acid insoluble or the retinate fraction. Please let me know if you have any doubts. Yes, children. Let me know, please, if you have any doubts. Yes. Shillong. Do you have any doubts? Did you copy everything? And yes, today we are not going to have any break. I'm just letting you know in advance. Not going to have any break. We'll end up the class at 5:30. So we are going to proceed without break and we'll finish it by 5:30. Yes. What are you doing, children? Did you copy? Did you copy? Okay, understood everything? Each and every child. How many of you are there at the center? Today you seem to be very tired. Is it? Huh? Today you seem to be very tired. Is it so? 7, okay. Are you tired today? You seem to be. Understood everything? All 7 of you. Each and every one among you. Clear? So what is going on in your school? In bio? What's your teacher teaching? Your chapter? Yes, but you don't seem to be. You seem to be tired today. Responding so late. You are responding, yes, but very late. I guess there's some signal problem or whatsoever. Might be. Yes. What's your teacher teaching you at school? Yeah, I guess. Might be. No issues, don't be sorry. I was expecting the internet to be the root cause. Yes, and what is your teacher teaching you? Yes. At school, what is being taught, children? Okay. Biomolecules. So now this is like, you're getting clear idea? You're having any problem? The idea is clear? Why did the lipids, they did not come in the filtrate part? Why were they left in the retinate section? Okay. Now, one more thing I would like to show you over here. See, if you just pay attention, you'll be able to see the amino acids. The amino acids are the monomers of proteins. Or other way around, proteins are polymers of amino acids. Monosaccharides are monomers of carbohydrates. Or other way around, carbohydrates are polymers of monosaccharides or the simplest carbohydrates. Nucleotides are the monomers of nucleic acid. Or nucleic acids are the polymers of nucleotides. So I will proceed this chapter in this way. I will be teaching you first amino acids, then proteins, Then we will do monosaccharides, carbohydrates, nucleotides, nucleic acids in this way. So what you have over here, you have a particular structure called as lipids. Now lipid, what to talk about this thing? It is a micromolecule or macromolecule? This is a micromolecule because its molecular weight is how much? Its molecular weight is only 18 to 800 Dalton. So what do we call lipids? We call lipids to be biomicromolecule. It is the only molecule which among these which does not have a micromolecule or which does not have a polymer. So amino acids, they have a polymer called as protein. Monosaccharides, they have a polymer called as carbohydrates. Nucleotides have a polymer called as nucleic acids. Polymers you know, isn't it? The building, like one, if you take into account one brick, it is a monomer. When you keep many bricks on a particular surface, they form a polymer called as wall. That is what is the building blocks. So monomers are repeated many times to form a polymer. So if you are repeating amino acids many times, what you are going to get? You are going to get a polymer called as protein. When you are going to repeat the monosaccharides many times, you are going to get a polymer called as carbohydrates. Same is with nucleotides also. But yes, lipids, they don't have any macromolecule. Just mention this over here only. Or we'll try to do this thing in this way. So lipids do not form polymers. Clear? Clear with this, children? Clear? Very good. So can I start with the first thing, that is amino acids? Can I start with amino acids? Starting with amino acids. Then we will do proteins. After proteins, we'll do lipids. And then we'll see what next follows. Okay. So you have copied this all. We'll start off with amino acids. So amino acids are a typical kind of molecule which has an amino group as well as an acidic group. Let's see its general structure. This is amino acid. This group is called as amino group. This is the carboxylic acid. This is variable, that is, it varies from one to another amino acid. So this carbon, the carbon which I have like drawn over here, is said to be alpha carbon. Alpha carbon is a carbon to which we attach the functional groups. Apart from being alpha called as alpha carbon, it is also called as chiral carbon. So chiral carbon is a carbon to which we can say not more than, but we have to say to which four different groups are attached. So if you are attaching four different groups, like number 1 is NH2, number 2 is H, number 3 is COOH, number 4 is variable R. So what we are doing is we are attaching different groups on different bonds to the carbon. So this kind of carbon is said to be what? This kind of carbon is said to be chiral carbon. And this is what carbon? This is alpha carbon. So alpha carbon is a carbon to which we attach the functional groups. So we have attached the functional group, we call them to be alpha carbon. And if all the functional groups or all the groups are different in the carbon attachment, we call it to be chiral carbon. Clear? This is the general formula of amino acid. Now this amino group, did you copy this, children? All of you. Did you copy this? Okay. So this NH2 group gives it variability. That is, this is the one which provides it basic nature. You know basic acidic, you must be aware about it. And the COOH gives it acidic properties. So, if the number of NH2 is equal to COOH, we call the amino acid to be a neutral amino acid because yes, both the things will be neutralized by the other component. We call the amino acids to be neutral, that is NH2 equals to COOH. Examples you can have is glycine, valine, leucine. These are the amino acids where we have equal number of NH2 and COOH group. Now, the other example, if NH2 group is more than COOH. Why? Because the R group, remember this R thing? This R thing. It might contain NH2 group. Now, if this R group is containing a NH2 group, obviously the number of NH2 will be more than COOH. These kinds of amino acids are said to be basic amino acids. AE is for amino acid. So neutral amino acid, NH2 equals to COH. Basic amino acid, NH2 is more than COH. The examples are lysine, arginine. These are the basic amino acids. Okay? Now, third, what if the R group has COH group? Obviously, the number of COH will be what? Will be more than the NH2. If NH2 is less than COH, this amino acid would be said to be acidic amino acid. And what examples do we have? We have is aspartic acid, and glutamic acid. Clear? So in this way we have three different kinds of amino acid. Aspartic acid can also be written as aspartate, and this can be written as glutamate. So either ways it is correct. Clear with this, children? Clear? So we have what? We have three different kinds of amino acids on the basis of number of NH2 and COH group. If the number of NH2 and COH group is equal, we call the amino acid to be neutral. If the R has NH2 extra, obviously the number of NH2 will be more than COH, called as basic. And similarly, if COH is more, if the number of COH is higher than NH2, it will be called as an acidic amino acid. Examples: glycine, valine, leucine, neutral, lysine, arginine for basic, and for acidic we have aspartic acid or aspartate and glutamic acid or glutamate. Clear up to this, everybody? Yes, Shillong? Can I move ahead? No doubts? Now, we will see the neutral amino acid. We will be exploring it to the further level. Next thing. There are some amino acids which have sulfur containing. They are sulfur. They contain sulfur. What are they? Cysteine, cystine, and methionine. These are three sulfur containing amino acids. Next we have is the ones containing OH group. It is hydroxyl containing amino acids. Serine, threonine. Next we have is what is aromatic? What is aromatic? Yes. What is aromatic amino acid? What is aromatic first of all? They have a benzene ring. So they contain benzene, that is the particular ring structure that you must have studied somewhat like this. See. Remember? You must have studied in this way or in the form of this way either. So this is what is benzene containing amino acids. What are they? Tyrosine, tryptophan and phenylalanine. So again, the neutral amino acids have been divided according to the functional groups. The first and foremost we have the ones containing sulfur, cysteine, cysteine, methionine. Next we have OH group or hydroxyl group containing serine and threonine. Aromatic or benzene containing tyrosine, tryptophan, phenylalanine. Are we clear? Okay. Can I move ahead, children? Okay, very good. Next what we have is, these particular amino acids, they have a property. They have a particular ion structure formation called as zwitterion. We'll write down as like this. Now what is zwitterion? Zwitterion is an ion having both positive and negative charges at the same time. So at the same time, at the same point of time, what they have is, they have both positive and negative charges. So they have plus and minus charges at the same particular timing. So they are said to be what? They are said to be zwitterion. So amino acids have a unique property of forming zwitterions. That is, they can carry positive and negative charges. I can see students sleeping. Yes, why are you sleeping? Get up everybody. Yes, very good. Okay. So amino acids are the particular ions which are having positive and negative charges at the same pH. Okay, we will see this thing. At the same point of time, at the same pH obviously. What is pH before that? What is pH? We have studied pH in digestion also. What is pH? We talked about pH so many times. But what is pH actually? Yes. What is pH? Yes. Potential of OH. Kind of, yes. Right? So this is basically what we define. Let's first see the definition. pH equals to negative or inverse log of H plus ion concentration. This box, this big bracket, if I am indicating this big bracket with H plus, that means it is concentration. So this particular thing indicates concentration. This particular bracket, this whole thing, This means concentration. Sir will be teaching you all these things. But yes, I will be just letting you know what is the basic thing, why we are doing this thing. So, what is pH? pH is the negative log of H plus ion concentration or inverse log of H plus ion concentration. So, in this ways, what a pH value is like, if you increase the H plus ion concentration, since it has an inverse relation, see over here, it has an inverse relation, isn't it? So, more is the H plus ion concentration, less will be the pH. Or, less is the H plus ion concentration, more will be the pH. Isn't it? This is what we know. So, we also know about a scale called as pH scale, where we have values from 0 to 14. And in the center we have 7. Anything which is having a pH of 7 is said to be neutral. And 1, 2, 3, 4, 5, 6. Okay. Anything which has a pH of this value, this is decreasing pH. Decreasing pH means the H plus ion concentration is increasing, isn't it? At the same time, from 7 to 14 is pH is increasing. Other way around, we can say H plus ion concentration is decreasing. So, if you have a pH of 1 and pH of 6, or let's say 4, which is more acidic? Out of this, which is more acidic? pH 1 or pH 4? Yes, what is more acidic, 1 or 4? I'm waiting, Shillong, tell me. What happens to be more acidic? Yes, very good. So pH 4 is more acidic than pH 1 is more acidic than pH 4. This phase. So, this pH concept is now clear to you, I guess. So, coming back to the main topic again, what I was talking about, back to biology. What I was talking about deuterions, that amino acids, they can form what? They can form deuterions. Okay, fine. So, what are deuterions? Quickly, let's see. So, this is an amino acid. So, if you are decreasing the pH, I am moving towards the left. Obviously, just now you said to me in the pH scale part that if I am moving towards the left, that is moving towards 0, pH is increasing, pH is decreasing and H plus ion concentration is increasing. So, what I am doing is, I am increasing the H plus ion concentration or I am decreasing the pH. So, this is a place where I have abundance of H plus. Okay, so if abundance of H plus is there, what the molecules will tend to do is that they will tend to donate something. What? They will tend to donate the H plus ion. So what they will do in the solution, this kind of solution is, they will donate the H plus ion to the missing things. Obviously, COO was minus. It was in lack of H plus ion, in lack of hydrogen. So this kind of solution is going to give it a H plus ion, H plus ion. Now, the next situation is, I'm going to increase the pH. Or other way around, I am going to decrease the H plus ion concentration, or I am going to increase the pH. So if I am going to increase the pH, means there is a potential where the H plus ion is less in this solution. Now imagine a situation, if you are moving towards a place, you are going to a place where there is scarcity of something. What they will do? They will try to snatch off the things with you. So this particular ion will lose its H plus in this solution, and this ion is going to gain the H plus in the solution, whereas this ion has both loss and gain. So this particular B ion, the B ion in this whole structure, is said to be the zwitterion. Why? Because at the same time it has positive as well as negative charges. Positive with NH3 and negative with COO minus. The idea is clear to one and all? Let me know if I need to explain something once again. Yes, Shalong. Clear? Everybody? Yes. Just give me a thumbs up if it's clear. We'll do some MCQs now. Yes. Quickly. Okay, very good. So we will have some questions. First question. Starting the questions. Yes. Answer these two questions. Quickly, Shillong. What is taking so long? I have to give more questions. You can just take a pic of this or you can just copy it later. First tell me the answer. Yes. Good. What about next? Number 2? Okay, let's see. This one? Yes. Number 3. Number 4. Yes, number 4 and 5. Very good. And 5th? Fifth one. Okay, let's match your answers. Let's see how much you have scored. So, the 88 part does not have what? Obviously, the 88 part does not have amino acids because it is present in the filtrate section, as well as it does not have nucleotides. So, it does not have either of them. That is amino acids and nucleotides. So, the correct answer is what? Both A and C. Very good. Cytosine is formed by which of the following molecules? It needs to be amino acids. Okay. So what have you answered? Amino acids. Very good. So both carboxylic and NH2 participate. Amino acid containing hydroxyl group. Just now we were talking about. This is sulfur containing amino acid. Does not contain any this kind of variation groups. This contains OH. This also nowhere. So what the correct answer is? C, 3C. Okay, very good. Doing good. Out of these molecules which does not form a polymer, obviously, amino acid forms protein, nucleotides form nucleic acids, monosaccharides form carbohydrates, and we have lipids as the correct answer because they don't form any polymer. I have taught you this thing. Fourth D, very good. Good going. And basicity, last question. The basicity of amino acid is due to, obviously, the NH2 group without any exclusion. We know that NH2 group provides it basicity. So how much you are going to get? Full marks. Very good. Keep it up. Okay, so we have done with the whole section of amino acids. In the next class, obviously the next week, I guess, we'll be getting a class. So in the next class, we will start up with proteins, the polymers of amino acids. So just let me know if we have any doubts, or shall I wrap up the class? Yes. Are we having any doubts? Everybody, children? No doubts? Okay, fine. Is it all clear? Shillong. Pack your bags later. Just tell me if everything is clear or not. I'm waiting for your reply. Okay, fine. Okay then. Crystal clear. Okay, fine. So we'll be meeting up in the next class and continuing with the proteins section. Okay, let's see how much we can cover in the next part. And yes, I have given you certain assignments today also. Don't forget to do that. That was from the last chapter, excretion. So you have to do that assignment. It's a subjective assignment. You have got whole 1 week to do that assignment. So utilize the whole time for that thing. And let me know if you have any problem. You can talk to me on the group also. As well as if we meet in the next class, you can talk to me there also. Okay, fine. So let's wrap up. Okay, good night. Thank you so much. See you again. Bye-bye. Take care.